I want to try to get the board and the middle. So if you're right, it's okay.
Got it?
You're gonna have to press play.
It's already going.
Oh.
Yeah, you press.
All right, so as we start here with the urinary tract.
Oh my goodness. That was my fault. That was my fault. Here we go. Okay.
Edit that out.
We'll edit it in post. It's fine.
Probably not, like that's relying on some editing skills there, whatever, we'll just go. All right, so as we start the urinary tract, one thing here is the kidneys are complicated. And there's a lot of like tiny details here, and there's a lot going on here that we're not even going to talk about. But what I'd like you to keep in mind as you go through here is, despite what anyone in nephrology will tell you, the kidneys are not the only organ in the body. Like, there's a lot of other things going on. So what we need to focus on here is the big picture idea of what the kidneys are doing and how they do it, not necessarily the minutia of how that's getting done. So some things we'll talk about in an inordinate amount of detail, but keep your eye on the bigger picture of kidney function, and don't get lost in some of the movements of ions and things. Some of them are important, and some of them we just need to understand the bigger idea of what's happening. All right, so the urinary tract, the kidneys, the urinary bladder. So the kidneys are really the only important part of this. The rest of it's just kind of a tube and a passageway out. So really, we're mostly worried about the kidneys. They're the excretory organs, and this is the major function of the kidneys, is excretion. That's sort of sorting through what you've got and getting rid of the stuff you don't want in your blood. And the rest of it is just kind of transport out of the body. So here's the kidneys. You've seen kidneys. They're the kidney-shaped things, and then against the posterior body wall there, little adrenal glands, like little party hats on top of the kidneys there, connected to the bladder through the ureters. The big function, like I said, of the kidneys is excretion. That's removing stuff you don't want out of the blood. Now, sort of as a consequence of that, the kidneys are also regulating blood volume, which means they're regulating blood pressure. They're regulating the chemical composition of the blood, so that means they're regulating the pH of the blood. So these are also functions of the kidneys. But big picture, excretion, filtering the blood out, getting rid of stuff you don't want. In prolonged fasting, the kidneys will do gluconeogenesis, which means you're making glucose out of stuff that's not. So, that's not one of the major functions, but it's there. The kidney does release renin, which is our renin-angiotensin system, which we spend a lot of time with cardiovascular. And it releases erythropoietin, which we talked about when we talked about blood production. It's also one of the steps in activating vitamin D. You've got the skin, and the kidneys, and the liver all working together to make vitamin D. You've seen where the kidneys are. The kidneys are actually retroperitoneal, which means they're behind the membrane that covers everything else, the peritoneum. Let's look. There's the kidneys, right up against the posterior body wall, partially protected by the ribs, and behind the peritoneum. We look at the kidneys. The outside of the kidney is called the renal capsule, and it's sort of the shell on the outside of the kidneys. And then around that, there's a fat capsule called perirenal fat, so fat around the kidneys. They're embedded in a wall of adipose tissue that's right up against the posterior body wall, and it helps lock them in place. And then there's this fascia that covers that. The renal fascia anchors the kidney. So here in this cross section, we can see the peritoneal cavity, and there's the peritoneum, this membrane that goes around, and the kidneys are behind that. And then there's this perirenal fat capsule around the kidneys. If we open up the kidney and we just put it in sort of a frontal section here, we can see these very distinct regions. So the sort of outer shell, like the brown outer surface, is the capsule, but then inside we have an outer layer and an inner layer. The outer layer is called the renal cortex, and the inner layer is called the renal medulla. It's really easy to see this distinction because the renal medulla is made up of things called pyramids and columns, and they're very obvious. So even if we weren't looking at like an illustration, so there's like the drawing of this thing, you can still see those pyramids and columns. So the renal cortex is this outer layer here, and then everything on the inside is the medulla. And then here's your pyramid, columns in between the pyramids. A renal lobe is a pyramid and the cortex above it, so that's a lobe. The tip of each pyramid here, that's called the renal papilla. And at the end of that, you have these structures that, like this. Each one of those is called a minor calyx. Calyx means cup. And urine is going to form in this direction, and it collects here in the minor calyx. And then those minor calyces here come together into this region. This is a major calyx. And then those major calyces drain into here. This region is called the renal pelvis. And then the renal pelvis drains that urine down into here, into the ureter. So like I said, these structures are really obvious even in the gross anatomy of the kidney. With a microscope to see those different layers of tissue. All right, so papilla, the tip of the pyramid, renal pelvis, see again, and then the minor and major calyces, urine going out into the ureter. The ureter leads into the bladder. The renal arteries, which we've already looked at when we looked at cardiovascular, right, left and right renal artery. The renal arteries take about a quarter of cardiac output in the kidneys every minute. Cardiac output, totally removed from the heart thing. Cardiac output, milliliters per minute, how much blood the heart's pumping out per minute. About a fourth of that goes straight to the kidneys. Arteries in, veins out. We're not worried about nerve supply yet. Here's our structure for these arteries. So what you see is the big renal artery coming in, and it branches off, and we have smaller branches. There's the big one, and it branches off. That's a segmental artery. The first branch off the renal artery is a segmental artery, and then that segmental artery branches off into smaller interlobar arteries. And those interlobar arteries come up here and they make this arc across the top of the pyramid. That's called an arcuate artery. And then off that arcuate artery, all these little bitty arteries that come out here in like little spokes, those are called cortical radiate arteries. Now notice that as we go through, we're getting smaller and smaller and smaller. And off those cortical radiate arteries are the tiniest of them, the little arterioles, which you can't really see in this picture. And those will lead into the capillaries. So here's our pathway of blood through the kidney, starting at the aorta. Branches off in the renal artery, segmental, interlobar, arcuate, cortical radiate, and here's where the action takes place, is down in these capillaries. And then drains back in the veins, which have virtually the same name. The action that's taking place here takes place in the nephron. The nephron is the structural unit and the functional unit of the kidney. There's at least 1 million of them in each kidney. This is where the kidney's doing its job. And there are two main parts here. The first part is called the glomerulus. The glomerulus is this spaghetti ball of capillary. If we enlarge it, it looks like that. So this is a capillary bed. But unlike other capillaries, it doesn't just spider web out and go straight into a vein. And instead, it's this ball of capillary. And that this is not a vein yet. We're not even to the vein. We're still in an arteriole here. So there's our glomerulus. And then the rest of it is the renal tubule, which is going to catch everything that comes out of the capillary. So here's the nephron. So now we see here's our, this is the arcuate artery, here's our cortical radiate artery. And then here's our arterioles coming into that and then coming back out. Then here's the tubule system. It starts as this sort of cup thing that's around that ball of capillary, and then we've got this system of tubes that leads into that big collecting duct. The renal corpuscle is the glomerulus, that capillary ball, and that sort of capsule around it. The capillary here is a fenestrated capillary. Remember that a fenestrated capillary has tiny little holes in it. And those tiny little holes lets fluid escape into that capsule. From the capsule, we enter the renal tubule. Now the capsule itself has an outer layer that's the parietal layer, it's just structure. The visceral layer is made up of cells called podocytes. Podocyte means foot cell. And podocytes extend out and they wrap around the capillaries. They create these little things that are little slits in between the cells called filtration slits. So there's the outer layer, here's the inner layer. Now what we see here is the capillary, and you can see the hole in the capillary there. It's a fenestrated capillary. Then we see the podocyte, this foot cell, and there's a little filtration slit right there. And their basement membranes are fused together here. So if we want to get something out of the blood and into this capsular space, this is the filter we're going through. We're going through there and out through those slits. After this, we have the renal tubule, where all this fluid that's going to get pushed out is going to be processed. The first part of it is called the proximal convoluted tubule because it's closer to that glomerulus. The proximal convoluted tubule is made up of cuboidal cells with lots of microvilli. Remember what microvilli do? Microvilli are there to increase the surface area. Anytime you see microvilli, you should associate that with absorption or secretion, or something's going to be moving across that cell. So in the small intestine, when we see microvilli, it's surface area so we can absorb stuff. Here it's surface area so we can reabsorb stuff. And we see lots of mitochondria. Remember what mitochondria do? Mitochondria make ATP. That ATP is for active transport. Surface area, passive transport. So these cells here in the proximal convoluted tubule are specialized for transport. We're going to be moving a lot of stuff from what we just filtered out back into the blood here. So here's a cell for the proximal convoluted tubule. See those microvilli, lots of surface area, crap tons of mitochondria, lots of ATP. From here, we enter what's called the loop of Henle, or more appropriately, this is called the nephron loop. And the nephron loop is going to go down into the deeper kidney and then come back up. So we have a descending and an ascending limb. And there's a skinny side and a thick side. A thin segment and a thick segment. The thin segment is mostly simple squamous epithelium and it's permeable to water. The thick segment is mostly cuboidal cells that's not permeable to water. So here's our thin segment cells. We're mostly moving water here. Finally, we go into the distal convoluted tubule. In the distal convoluted tubule, the cells look like the proximal convoluted tubule cells. They're cuboidal cells, but there's not as many microvilli here. We don't need the surface area because we've already processed all this fluid, so we're not doing a lot of passive transport here. There are a lot of ATP for active transport. And there's our distal convoluted tubule cells. The last part of the nephron is called the collecting duct. The collecting duct is getting the stuff that got filtered out from all these nephrons, and this is your last chance to sort of process this fluid. What's left will be urine. So there are a couple of cells here in the collecting ducts. We have what are called intercalated cells. These are cuboidal cells, lots of microvilli, and they're going to be involved in acid-base balance. They're moving different ions across for this acid-base balance. There's a intercalated cells there in the middle. The other cells around it are called principal cells, still cuboidal cells, but really no microvilli, and they're involved in water and sodium chloride balance, basically. Talk more about function in a bit. All right, so again, our nephron. And this diagram of the nephron is very simplified because there's like a million of them in each kidney. They're overlapping each other, they're on top of each other, they're next to each other. Like there's multiple nephrons coming off that one cortical radiate artery. We just simplify it so we can just look at the one nephron. Most of your nephrons are in the outer layer of the kidney, the cortex. About 85% of nephrons are nearly entirely in the cortex. They're called cortical nephrons. About 15% of the nephrons cross deeper into the medulla. They're called juxtamedullary nephrons. And these will be more important in concentrating urine. So here we see our cortical nephron, and you can see that loop just kind of goes slightly down and up. And here's our juxtamedullary nephron, where that loop's really long and goes much deeper into the medulla. One thing to notice in this picture are these purple capillaries here. So there's your glomerulus and that little capsule. Here's the arteriole going in, and there's the one going out. And you can see the one goes out leads into this peritubular capillary. Okay, so let's talk about that. The artery, the arteriole that's going into the glomerulus is called the afferent arteriole. Afferent always means in. Efferent means out. So you have an arteriole going in, it sort of spaghetti bowls up in this finished end capillary, and you have the efferent vessel going out. This is filtration. This is where the filtering part happens. Glomerulus, filtration. That's where filtration takes place. The reason this works is because of pressure. Because that afferent arteriole is bigger than the efferent arteriole. The one that's going in is bigger than the one going out. And what that does is it puts pressure in the middle, and it shoves fluid out into that capsule. So if we've got this bigger vessel that comes in, and then it does that whole spaghetti roll thing, which I'm going to draw expertly here. And then you've got this smaller vessel going out, all that pressure in the middle. Fluid is going to move out here. So fluid is moving into the capsular space. All right, that means that blood's still circulating through, but now the blood that's coming out here, there's less fluid in that blood. So what's the pressure like? If I reduce the volume, what happens to the pressure? I reduce the pressure too, right? Volume pressure directly correlated. So reduce volume, reduce pressure, and this efferent arteriole. That's important because that efferent arteriole leads straight into those peritubular capillaries, the capillaries that are around the tube. They don't have a lot of pressure in them. That's great because fluid wants to move with a pressure gradient. The pressure is, and let's go back. The pressure is inside this tube, the yellow tube here that's got the fluid in it. And then we get this capillary wrapped around that doesn't really have any pressure in it. So fluid is going to move from that tube back into the capillary, into the blood where it needs to be, as it's filtered through this tubule system. The vasa recta is that really long purple capillary that's going into the juxtamedullary nephron. Same principle though, low pressure coming off that efferent arteriole. We talked about net filtration pressure before. The net filtration pressure here is different. Because of the way the efferent and afferent arteriole are, there's a lot more pressure in the glomerulus than we have in other capillaries. So it's important that we reduce blood flow going into the kidneys, especially into that glomerulus, because higher pressure would cause it to explode. So the blood pressure goes down tremendously inside the kidney because of resistance, because we reduce the amount of blood flow. This protects the glomerulus from fluctuations in systemic blood pressure, so that when your blood pressure goes up for whatever reason, it doesn't rip the kidneys apart from the inside. And the efferent arteriole, it keeps the pressure in the glomerulus, and it lowers the pressure in those peritubular capillaries so we can reabsorb this fluid. Every nephron gets this thing called the Juxtaglomerular apparatus. These are modified parts of the nephron, and they are there to modify the function of the kidneys, sort of on a real-time basis. Some of these cells are called granular cells. Granular cells are in that afferent arteriole. They're smooth muscle cells, and basically they detect blood pressure. Remember, your kidneys rely on blood pressure to do their job. We talked about this when we talked about blood pressure. The kidneys require this blood flow, they require this pressure to do their job, and if they can't do that, they release renin. Renin comes from granular cells. So if there's not enough pressure in that afferent arteriole as blood is coming into the glomerulus, the kidneys can't filter the blood because you don't have enough pressure to shove stuff out. So you release renin, and renin kicks off that renin-angiotensin system that we've talked about. The other part of the juxtaglomerular apparatus is the macula densa, and these are part of the ascending nephron loop, and these are chemoreceptors. They detect the salt content of the stuff that you filtered out. What you'll see is that as fluid moves through the tubule system, we reabsorb the vast majority of stuff that we threw away. Like most of it's coming back into the blood. If stuff is moving to the kidney too quickly, then we don't have time for that. And these receptors detect that sodium content. And if stuff's moving too quickly, they are aware of that and they slow the process down. And we'll look at that process in just a bit when we talk about autoregulation. There are mesangial cells that connect the macula densa and the granular cells, because both of these things are involved in how quickly the kidneys filter the blood. So more on this juxtaglomerular thing in just a bit. But here's our juxtaglomerular apparatus. So there's our modified cells inside this afferent arteriole, and here's our mesangial cells, and then those macula densa cells in that ascending loop. To get from the blood to the filtrate, we have to pass through the filtration membrane. Just like to get from the lungs to the blood, we have to pass through the respiratory membrane. Here we have the filtration membrane. This is the membrane between the blood and that capsule around that glomerulus. So 3 parts: the capillary, the podocyte, and their membranes that are fused. So here's our filtration membrane. You can see the capillary. Here's these podocytes wrapped around it, and there's a little filtration slits. When we want to get from the blood to this space out here, you've got to go through this filter. That's the filter. All right, so what can fit through the filter? Most stuff. Blood cells can't. Blood cells can't get through. Proteins can't get through. Most big molecules can't get through. Water does. Electrolytes do. Simple sugars do. There's lots of stuff that does go through. But the big things can't. All right, those mesangial cells will help break down anything that's caught there in that filter. So here again is our filtration membrane. Go from the plasma to this capsular space. The kidneys filter your entire volume of blood roughly 60 times a day. What we're calling filtrate is basically plasma minus the protein part. How much of that do you lose? Less than 1% of it. If you were to lose it all, you would pee out the entire volume of your plasma within an hour. Like it'd just be gone. So we reclaim most of the vast majority of the stuff that we filter out, we put it right back in the blood. Less than 1% of it is lost. And what should be there? Stuff that we didn't want and water. Everything else gets to go back in the bloodstream. So let's start with our mechanisms here of forming urine. Filtration, reabsorption, and then secretion. Okay. So, you ever seen that show Hoarders? If you've never seen Hoarders, if you need to feel better about yourself, Hoarders. If you're unfamiliar with this, Hoarders explores a psychological disorder, hoarding, where people, they don't, collecting is not the right term for this, where they keep everything. And sometimes they go out like actively to keep other people's things too. They don't throw trash away quite often, they just compile this. There's some really, really bad ones, like where the lady's got like 40 cats in her house, and that's not counting the dead ones in her freezer. There are people that like bottle their own urine because they don't want to get rid of it. They might need it later, you never know. There was one the other night where the dude had like 1,500 rats in his house. They were his pets. And he just like, at some point he's like, I didn't have enough cage, so you just let them free range. Oh my gosh. Like I think they ended up, that was one that actually had to like tear down his house. Because they had just eaten everything in the walls. So if you've ever watched this show, like the first thing they do, like they go out and they talk to this dude, like there's some psychological help. And so like he's got all this crap piled up. You can't even walk in the house if there's a narrow pathway, maybe. Like if the sack of phone books falls on this guy, he's gonna die. But he's like, I might need the phone book. And it's all piled up. So the first thing they do is like they talk to this dude, they're like, we're gonna do this, we're gonna help you, but we gotta get your house cleaned out. And so they bring in a crew, often a hazmat crew, because of, various dead animals and such. And they move everything out of the house. Everything they can. Everything that will fit through the doorway, they move out. This is exactly what's gonna happen at your kidneys. At your kidneys, your blood's going through and it's got all this stuff. It's got ions and it's got, carbohydrates and stuff. And the first thing your kidneys are gonna do to fix this problem is they're gonna move everything out. And then they're gonna sort through it and see what we actually need to keep. And this is the same thing that happens. And they don't make this dude throw everything he's got away. They're gonna go back through your stuff. They're like, oh look, family photos, baseball cards from the 30s, you're a millionaire. Like, you had no idea that you had all these gold bars. This sort of thing. And they're like, we should move that stuff back in the house. But like the big appliances, like there's a refrigerator, there's a freaking grand piano in here that you didn't even know you had. They're not moving that stuff out the door. And this is the same thing that's gonna happen to your kidneys. The big things, they can't even go through the filter. So they're gonna stay inside the blood. The rest of it, we're gonna shove all that stuff out and then sort through it and see what we really needed, what you really wanna keep. And then we'll move all that stuff back in. So this is the process. Filtration, we move everything out, and then we reabsorb most of it. Put it back in, because we actually needed that. We just needed to be organized better, I guess. And so move it back in. And then sometimes, like, again, when you watch the show, like sometimes you go back and they're moving stuff back in the house, and that guy's got like one dead cat under his arm and he's trying to sneak it in. And they're like, no, let's get rid of that. And that's the last part of this. Secretion is going back through and saying, actually, no, let's throw that away too. So this is the process. Sorting through the bloodstream. And here's where it's going on. Filtration at the glomerulus, the rest of it in that tubule system. Whatever is left over is what's going to become urine. Past the kidney, it's urine. Like once it gets out of the tubule system with the nephron, urine. There's no reclaiming that. Like if it goes in the urine or in the bladder, it's gone. You can't reclaim anything once it's there. Okay, so first, filtration. Sorting everything out, moving everything we can out the door. This is driven by pressure. It's a really good filter because we have that pressure in the middle. We've got that net filtration pressure pushing out. It's a fenestrated capillary that's super permeable. And there's lots of surface area there because it's like a spaghetti ball. And you get those podocytes wrapped around it. Again, things that are bigger than like 5 nanometers can't go out. That's actually useful because that also means that we don't shove all the water out of the blood. The stuff that stays behind maintains the osmotic pressure of the blood. So that water, some of the water at least, stays inside the bloodstream so that we keep this fluid and we keep it moving. So net filtration pressure, we've already talked about somewhat. Here, this is the pressure that's responsible for filtering the blood. Now it's higher than it is in other capillaries. And the reason for that is because that afferent arteriole is bigger than that efferent arteriole. Just like it was with the other capillaries, this is the sum of all the pressures here. We have hydrostatic pressure, this is blood pressure. Hydrostatic pressure, blood pressure, out. This is what's pushing stuff out. But just like on other capillaries, we also have osmotic pressure, the stuff that's trapped inside the blood, the big proteins and stuff. This is pulling stuff in. And because we're pushing this fluid out into a capsule and not just in the interstitial space, it's going to build up around the capillary, and this pressure that builds up out there is also going to push stuff back in. And when we add all that stuff together, we get the net filtration pressure. It's about 10 millimeters of mercury. So here's this. So here's our afferent arteriole, the one that's coming in. Pressure here in the middle, and it's shoving stuff out. It's about 55 millimeters of mercury here, shoving stuff out. But then all those proteins that are trapped inside the bloodstream are going to pull water back toward them. That's osmotic pressure. That's sucking stuff in. And as fluid builds up out here in the capsular space, it starts to push back on that capillary. And that's going to shove some back in too. So what we're left with is about 10 millimeters of mercury pushing fluid and stuff out of the bloodstream and into this capsule that's wrapped around this capillary. So all that fluid's going to get caught here, and it's going to drain down into the tubule system, where it gets processed. How fast does this happen? Well, it varies from person to person. The glomerular filtration rate is the rate of filtration, and it's how much filtrate is formed by the kidneys in a minute. GFR is like 120-ish. Clinically, like if we do glomerular filtration, we're looking for better than 100. Below 100, you're kind of concerned. It does get less as you get older, like under 30s, renal failure. Under under 100, you were like under 60, like that's a bad sign. So this is something we care about, and it's something that and we'll talk about how we clinically determine GFR later, but the kidneys are monitoring like really the filtration rate here, and this is governed by some different things. One of them is pressure. That one's easy. If you push more pressure through there, you filter more fluid through, you push that fluid through faster. But there's some other mechanisms in play here too. So first we're going to talk about the intrinsic controls. This is the kidney managing itself. And then we'll talk about the extrinsic controls, which is the nervous and endocrine system playing this as well. Intrinsic controls, if your mean arterial pressure is in that normal range, and really even outside the normal range, the kidneys sort of manage their own thing. And there are two types of autoregulation here. The myogenic mechanism, that's smooth muscle in the artery constricting or dilating, and then the juxtaglomerular mechanism. The myogenic mechanism is easy to understand. If you increase the pressure on that artery, this afferent arteriole, the one that's coming into that spaghetti ball, if you increase the pressure on it, well, what you would do is you would force more fluid out now because the pressure's higher. And so that net filtration pressure would go up and you'd shove more fluid out. And if more fluid comes out, it's going to move through that tube really fast. So we don't want that. So if you increase the pressure on that afferent arteriole, it constricts. And that reduces the amount of blood that's going through it, so it slows down that process, and you don't get as much pressure in the glomerulus, which helps protect the glomerulus, and it slows down how much fluid you're shoving out, because now it's a smaller thing coming in. If you drop the blood pressure, the opposite happens. You drop the blood pressure, there's not enough pressure going in, so that arteriole will dilate and it'll get bigger. So now there's more blood going through, pressure in the middle comes up again, and you shove stuff out. This is normally what's running the show. If we increase the filtration rate, we increase the amount of filtrate that we form, and we can't reclaim everything that we need to. Most importantly, the salts. And what happens is that sodium chloride concentration is so high because you don't have enough time to reabsorb it, that it triggers those macula densa cells in that juxtaglomerular apparatus. And those macula densa cells send a signal that causes that afferent arteriole to constrict, which reduces your filtration rate. Both of these mechanisms are here to keep that filtration rate in a normal range. So the pressure goes up, the pressure goes down, we manage that, and we keep the filtration the same. Under normal conditions, this is all we're doing. Now, the nervous system and the endocrine system also have some say in this. When you're stressed, fight or flight response kicks in. Remember our fight or flight response, the sympathetic nervous system, we're rerouting blood to the heart and the skeletal muscle, not the freaking kidney. So what happens is the renal blood vessels constrict, which would decrease your filtration rate. Your kidneys hate that idea, and the kidneys release renin. We get a renin-angiotensin mechanism that kicks in. Blood pressure comes up, blood flow to the kidneys comes up. So we've got this sympathetic response that's happening here. Here's our renin-angiotensin system again. We've already talked about this. There's renin catalyzing that conversion of angiotensinogen to angiotensin 1. That ACE converting it to angiotensin 2. Blood pressure comes up because angiotensin 2 causes blood vessels to constrict. And it causes the release of aldosterone. And it causes the release of antidiuretic hormone. All of those things increase blood pressure. What triggers this? Well, if the kidneys don't feel like they can do their job because your blood pressure is too low, they release renin. If the kidneys feel like there's not enough filtrate being formed, it triggers this and they release renin. If adrenaline is dumped into the system, it binds to those beta-1 receptors, they release renin. And all of those things raise your blood pressure so the kidneys can keep doing their job, which is filtration and excretion. Okay. Don't worry about prostaglandin. After we shove all this out, we've got to get it back in. Because we moved everything out, and as it turns out, some of it's garbage, but a lot of it's stuff that you want to keep. This is reabsorption. Now, reabsorption is selective. We're not going to keep everything. Your blood doesn't want to be a hoarder, so some of it we're throwing away. All the organic nutrients that we just shoved out, we reabsorb that. So, amino acids, really what I mean here, our important one at the moment will be glucose. We need glucose, we like glucose, we burn glucose. We just shoved it all out of the bloodstream into the filtrate. We want that back. We're gonna need that. So we're gonna reabsorb that glucose, all of it. The water and the ions, this is regulated by hormones, by gradients. And to get back into the bloodstream, we're gonna have to go through those tubule cells, or at the very least around those tubule cells. So there are two ways to get from the stuff that we just threw out back into the blood, either through a cell or around them. Transcellular or paracellular. Transcellular means we go through a cell, in one side, out the other, back into the bloodstream. Paracellular means we sneak in between the cells. It has to be something really small, like an ion or water. So here's our transcellular route going through the cell, and then here's our paracellular route kind of sneaking in between them. Now, transcellular can either be passive or active transport. Stuff can move with a gradient depending on its solubility and whether there's transport proteins there. Or transcellular active transport, we're actively pumping something across that membrane and back into the blood. Okay, things we need to reabsorb. We need to reabsorb sodium. We need to get rid of potassium, we need to reabsorb sodium. So how's it gonna work? Well, primary active transport. Let's go, okay. So sodium is going to be coming in here into the cell. Why? Because there's a crap ton of sodium out here. And there are ion channels for sodium on this side. So sodium's gonna come into the cell. What happens to that sodium? Well, on the other side of the cell, we have our sodium potassium ATPase pump. We got a protein there. It's gonna pump 3 sodium out. And in this case, that out is back into the bloodstream and 2 potassium back into the cell. Active transport, so we're shoving that sodium out and back into the blood. What's gonna follow the sodium? What always gonna follow sodium? Water. So the water is gonna go over there because we shoved all the sodium back over to that side. So we're also reabsorbing the water at the same time. Other things that are out here. Glucose. So one of the ways that glucose gets back in is we use our sodium glucose symport. And we've talked about this as well in A&P2, where sodium and glucose can come in if they come in together. That's secondary active transport. It relies on the gradient from primary active transport to pull that glucose in with it. And then that glucose is going to get pumped out the other side back into the blood. Here's the thing. All right, reabsorbing sodium, awesome. As we do this, and we're looking at this idea, so we have our filtrate here. Here's that cuboidal cell. We've got glucose, we've got sodium coming in that symport, so that it's here in the cell. And then it's gonna get pumped out the other side into the blood. Well, that sodium glucose symport is a protein. It's a protein in the membrane. Like anything, there's a finite number of these proteins, right? You don't have all the time in the world because fluid is moving as it goes through here. So in essence, those proteins have to grab that glucose and sodium and get it back in the cell. And like anything, it's a protein. Proteins bind to it, it sticks, it changes shape, it comes in the cell. There's only so many of them. So there's a maximum amount of something that we can move back in the blood. It's called the transport maximum. The transport maximum is the most of something that we can move back into the blood from the filtrate. Normally, when we talk about transport maximum, what we're talking about is glucose. If you ever see the old I Love Lucy show? If you haven't, shut up. So if you've seen this, like there's one episode where they take jobs at like a chocolate factory, and it's classic. So there's a conveyor belt with these chocolates, and they're supposed to like grab them and wrap them, and it's this assembly line, and there's just 2 of them, and like these things are going by really fast, and they can't get them all. They're like falling to the floor, and they're trying to wrap them and failing because they're just moving so fast. There's a maximum number that they can do. And if stuff speeds up, you can't get that. If there's too much of it, there's no physical way to do it. That's what's happening here. Those little proteins are grabbing on the glucose as fast as they possibly can. But if you've got too much glucose out here in the filtrate, there's no way to get it all back in the bloodstream. And that's what we get in diabetics, is the blood glucose is so high that it all gets shoved out into the filtrate, and there's so much glucose in that filtrate that we can't possibly get it all back in the blood. Those proteins are grabbing it as fast as they can and putting it back in the blood, but they can't get it all. So if you saturate those molecules, those proteins, the rest of it ends up in the urine. Because this reabsorption of organic nutrients, this is taking place in the proximal convoluted tubule. The very beginning of this, the very beginning of that tubule system, you get all the organic nutrients back. If you don't get them back, if they're still stuck out here in the filtrate, they're gone. Because past the proximal convoluted tubule, there's no mechanism to get them back. We don't have any more of these pumps anywhere past the very beginning. It's very effective for people that are not diabetic. When your blood sugar is not 600, then yeah, we can reclaim all that. For people that are diabetic, that glucose stays in the filtrate and it's going to end up in the urine, and you'll have glucose in your urine. It's also going to cause osmosis, because now we've left something dissolved out here, so it becomes an osmotic molecule and it pulls water back into the filtrate. So diabetics, one of the symptoms, you got to pee all the time, because that glucose is an osmotic diuretic. It's pulling water into the urine because you couldn't get it back into the blood. Okay. So water, osmosis, because it's following these dissolved molecules. Anything that's fat soluble, we're not really going to talk about, because things that are lipid soluble freely diffuse through the membrane anyway. We don't have to worry about that, it's just straight up diffusion. So here's your stuff. So there's our glucose and sodium coming in through the stem port. There's our sodium getting pumped back out. Glucose will do the same. Here's water going straight through, following that gradient. Remove all the sodium over there, move all the glucose over there, so water goes too. And then here's like other stuff. Things that are lipid soluble will go through the cell. These small ions will go back into the blood. Now sometimes we reabsorb stuff that we really should get rid of. We'll deal with this in just a second. Like if like there you see like the urea moving back into the blood, well that's something we'd kind of like to get rid of. And potassium too, we'll take care of it. What's stuck out here? And I tried this waste products that we need to get rid of. Potassium, urea, some other things will be stuck out here, and it's going to continue into the urine. So like I said, the proximal convoluted tubule is where most of this takes place. 65% of the sodium and water get reabsorbed in all the organic nutrients. All right, this is actually where we're going to stop for now. So we'll pause here, and on Thursday we'll come back and we'll look at the remainder of urine formation, and then look at clinical stuff after that as well. So take a short break and we'll come back and we'll look at the structures on the kidney board.